Okay, so this one's going to be nucleic acids. These are going to be some of your big molecules with a big role. So these are going to be the components of DNA, nucleic acid. And this is going to be found in your nucleus of your cells. It'll contain genetic information. Please try to not speak over me. It makes it very difficult for me to have to lecture. So, of course, this is DNA, deoxyribonucleic acid, DNA. And so the nucleic acid was right in the name of it. It's going to be what your genetic information is contained within. Typically, it's called your genome. Your gene is going to contain information to make the particular proteins of your cell. And so it codes all the RNA, which then gets going and gives you your proteins that your cells need and your body needs. The human genome contains about 20,000 genes. Your different cells or types are going to express different parts of their genome. And so when you're looking at a body, what you're going to see is that your DNA actually has different parts of it it's going to use depending on what cell you're looking at. Okay? And so we express different parts of your genome. All right? Nucleic acids, like DNA and its counterpart, ribonucleic acid, RNA, there's going to be very, very big molecules, especially DNA. They're going to have made up of repeating blocks. These are going to be called nucleotides. These are the components of your RNA and DNA. RNA being ribonucleic acid, DNA being deoxyribonucleic acid. Okay? DNA is slightly less reactive than RNA. And so it's a little more stable. If we're exploring the structure of our nucleotide building blocks, it's a starting point for understanding your structure of new nucleic acids and genomics, the study of genes and their interaction being genomics, of course. Just as proteins are going to be string of your amino acids, they're going to be these long polymers. The nucleic acids are going to be strings of molecules that are called these nucleotides. And so these are going to be the nucleotides right here and their components. So nucleic acids are created from a set of 4 nucleotides in a given sequence. Those are the ATCG, right? Everybody's heard of that before, right? Back in primary middle school? Okay. Yeah. No, maybe? Okay. And so nucleotides are going to have 3 basic components that they're going to be composed of. There's the base component. This is a nitrogenous base right here. Remember those heterogeneous nitrogen rings? That's what those are. Those are actually what tell you whether it's an A, T, G, C, or it's RNA, U. Okay. The next component is the 5-carbon sugar right here, and that's where the ribo comes from. So that ribonucleic acid, that's ribose. Okay. That's the sugar right there. And then this right here is your phosphate. And all together, when we assemble this, remember the PO4s we've been talking about, that phosphate anion? That's that. That's the phosphate backbone for your DNA. This right here is actually what is referred to as the nucleotide. You assemble these together with these phosphate groups, and that forms the DNA or RNA. Okay? And so that's the monomer for the polymer we know as DNA. Now, this base right here is the only part that differs in terms of your ATGC. This right here is the only part that differs in terms of your RNA versus DNA. And then U versus T, the base differs. Okay. DNA has T for that base. RNA has U for that base. Okay. Uridine. You may notice this looks a lot like something else we talked about, which is ATP, doesn't it? And so ATP is just adenine right here for the base. It's usually we're talking about the ribose one, so it's not the deoxy, but it can be the deoxy. And then we have 3 phosphates here. That's it. And so ATP is literally just the components, one of the components that would make up your DNA. Okay? In its most energetic form. These are going to be the nitrogenous bases right here. there's going to be 4 different nitrogenous bases found in our nucleic acids for our DNA. Each of the bases is going to have one of 2 nitrogen-containing aromatic rings. Will either be a purine or will be a pyrimidine. Okay. Whenever you're putting these together, these always need to be equally spaced across. And so what you end up with is the pairs. AT is a pair of purine and pyrimidine. GC is a pair of purine and pyrimidine. Okay. And so it gives you one that's a bit longer, one that's a bit shorter, and they click together. Okay. That's one of the checks that keeps them from mispairing. All right, and so right here, this is the DNA versus RNA. Your DNA and RNA are going to contain 2 purines. You have your adenine and your guanine. And these right here are your purines, adenine, guanine. Those are the same for DNA and RNA, no difference. Okay. The next one, there is a difference, and that's that DNA contains 2 pyrimidines again. They both contain 2. Thymine and cytosine are going to be your pyrimidines. Those are the 1-ring ones. Okay. The RNA is going to not have the thymine. It's going to have uracil instead. And that's where that U comes from. Is the RNA has uracil instead of thymine. Okay. All right, and those are the structures. You can see right here that these actually have very, very similar structures, don't they? Yeah. And so they actually do the same hydrogen bonding pattern up here. And that's why they replace each other. Again, this one being DNA, this one being RNA. Okay, let's look at the ribose versus deoxyribose. So this one right here is ribose. That's the R in ribonucleic acid, RNA. This one right here is deoxyribose. We can see right there, that position. We lost an OH. It makes it slightly more stable. Okay? It's less reactive. All of your structures in your body, by the way, are constantly being hydrolyzed by the water in your body. And so everything in your body breaks down over time. Okay? If you were to stop making new stuff in your body, you die. And that's pretty much true of all life. If you stop making new, you die. Extinction, gone. Or death, gone. Okay? And so in the case of DNA, that actually lasts for quite a while. You can usually last a few years with the DNA molecule on average. If we're talking about something like ATP, those only last a few days, actually. You'll have most of the population of ATP gone within a day or 2. Yeah. So if you don't make new ATP every day, you fade away really fast. Let's talk about the condensation of these components, of course. We have the pentose nitrogenous base. These are going to be joined with condensation reactions. This is going to be this nitrogen right here, bonding to the C1OH right there. And notice that's a glycosidic bond right there, isn't it? Okay, and it's condensation because we lost the water there. If we look over here, we can see R. What do you think that is? It's the condensation reaction, right? Okay. Okay. Anybody have any questions about our condensation reaction? We've assembled our base onto then our sugar using that wonderful glycosidic bond, just like we used for our sugar structures. And so what we have now is the other side is where you're going to react the phosphate. Again, this is going to just be another condensation reaction. And this is going to put that phosphate on this 5' carbon on your sugar. Notice that's the same carbon as you'd have for the alpha beta. This one was in beta linkage, wasn't it? Okay. And so once we assemble everything together, this is our DNA and RNA nucleotides again. A quick note about the naming. So a nucleotide is going to contain nitrogenous base, a sugar, and a phosphate. It has everything together. The nucleoside contains only the sugar and the nitrogenous base. So think of it as side of tide, right? Tide is the whole monomer. Nucleoside is just the sugar base. Doesn't have the backbone. Okay? So it's the side off the backbone, right? Okay. And there's our different ones. Notice, by the way, UMP right here. The U is uridine, M for mono, and then phosphate. Okay. Notice AMP here. That's the monophosphate. If we had triphosphate, that would be ATP, right? For the RNA, it's just the AMP. For the DNA, we put a small d in front of it for the deoxy form. Okay? And so it is small d, AMP. If we were the energetically higher energy one, before we would assemble these, that would be small d, ATP. Okay? Okay. By the way, what do you notice about this? We have ATP. Do you think we have a GTP? CTP? TTP? Yes. Yeah, we have all of those, okay? So you can have all of these as the triphosphate forms. Whenever we say ATP in any sort of energy thing in biology, if you're ever going to biology, they actually mean any of it. It doesn't have to be ATP. Okay? It could be CTP. It doesn't actually matter. They have the same energy. Okay? The mono versus the triphosphate has essentially the same energy. If we're naming the nucleotides inside a nucleoside name and the number of phosphates present, the nucleotide names are often going to be abbreviated. The abbreviation is going to indicate the type of sugar, whether we have the ribose or deoxyribose, and the nitrogenous base as form. If the deoxyribose is found in the nucleotide, we're going to have a lowercase d to tell us that. If we don't have the deoxy, and it's just the regular ribose, then we don't need anything else. And so we don't bother labeling. And those are the RNA ones. These right here are the names and the nucleoside that they would have. And that sums that all up. Many of the nucleotides are going to be linked together to form our nucleic acids. So nucleotides are going to be linked through what's usually referred to as a phosphodiester bond. And right here we can actually see the phosphodiester. Notice you have a phosphorus, so phospho, di meaning two, and esters were carbon O carbon, right? And so it's just the same sort of naming as we got in the IUPAC, wasn't it? Isn't that nice? Everything's consistent. Okay, again, the oxygens in the phosphate are going to be connected between the 3' right here on that one, and then the next 5'. And so whenever we read a DNA or RNA, we always refer to the ends as 3' and 5'. Okay? And that tells us which side we're on. So the nucleic acid sequences, these can be designated by one-letter base abbreviations. We use GCAT, right? And these nucleic acid sequences, one-letter abbreviations, of course, we're always going to go from the 5' end to the 3' end. And this will be our phosphate backbone, which is going to be consisting of the phosphates and the sugars. And so that's the phosphate backbone. The bases are then going to be jutting out from that. And those are going to be our GTCA. Okay? We can shrink that down a bit, in which case we go ahead and shrink it down. This would be a smaller writing. This is typically how we would actually list these if we were to try to list them. Though, frankly speaking, you don't actually see that often because DNA is long. Okay? DNA is very long. Even as one-letter abbreviation, it's very long. You always write it 5' on the left, 3' on the right, and by convention, you read it from the 5' to the 3' end. Okay? Because typically, it's going to be added to on this end. Okay. All right. We will cut off right here, and we'll continue talking about DNA and RNA. You all have a wonderful time. I will see you next week. Have fun. Don't forget, it is what for this weekend? It is Easter. Good Friday. You got Friday off. Have a fun weekend. Be haunted by I stopped on DNA. Now you don't know everything about DNA. Okay. You all have a wonderful weekend.